Okay, so we are back on track. So I was speaking about randomness and pattern and how, uh, you know, things appear random until we find a pattern in it. For example, if you look at the coastline of Norway, it's kind of random. But if you draw the map, the thing that you see is now the map of Norway. It's not random anymore. It becomes a pattern. And biology is about the process of uncovering those patterns out of randomness. In a way, the two extremes on the side, patterns and randomness, they're the most easiest to treat with. What lies between them is the hard stuff. Here, for example, you can see like the denticles on top of a shark embryo. And so we're slowly uncovering patterns. So here you can see that there is some repeating pattern of these denticles. And there's a lot more to be learned about sharks as we go on. If you look at this image, there are 5 pictures of 5 different embryos. And if you were to ask a random person, they would say that they all belong to the same species, probably even siblings or something like that. But if you look at the further development of these embryos, you can see significant changes. So this is where like the term, when people say that we share 99% of our DNA with chimpanzees come into picture. Like you follow some specific template and then you see deviations because of the small changes in your DNA. And we are all not random beings. For example, if someone were to see me, they would associate me with the pattern of Harish, for example. But I'm also not just a Harish pattern on the macro scale. You can find patterns within me at different scales. So you can, for example, you can look at the bones here and immediately you can figure out that this is probably a human skeleton. It's a pattern associated with humans. And you can have the pattern for the blood system, and you can go much shorter in scales. You can go to the scales of organs and tissues and cells and DNA and so on. And we're not just talking about patterns spatially, but rather also temporally, because, for example, our heart beats periodically. We know that the pattern is going to repeat and is going to come again. Essentially, the big question is, how are these patterns emerging? And one way to create those patterns is the cells that make up these patterns, they have to move, and they have to move collectively. And there are many major categories of cell motion, but I would start with like just 3 of them. The first one is morphogenesis, which essentially means formation of pattern. On the left side, here you see the elongation of an embryo. And this is like initially you're just a spherical ball, like most of the mammals, but you start elongating, you start breaking symmetry. And this is where you see that the cells are collectively moving and then folding. You also see that cells collectively move downwards and buckle on the bottom side. And on the top side, you can see cells creating the first hole inside a salamander embryo. So all of these features, they involve formation of pattern, which relies on collective motion of cells. When cells move collectively. This is very different from passive motion. So there's nothing pushing or pulling individual cells here. It's actually all the motion that's being seen is being performed by the cells themselves. Cells can also detect change in topology. For example, you have in normal skin, you don't have any holes where you can let water or other particles enter in. And if you have a hole, there is a wound, the cells can detect that and collectively move to change the topology and seal the wound. Collective motion of cells is also essential when you think about cancer. Here you can see a single cancer cell migrating, but the spread of cancer is more enhanced when a lot of cells coalesce into one small cluster, and the whole cluster moves to this new region, and then it breaks apart and starts invading. So collective motion cells is also essential when it comes to diseases. And to study this collective motion, we have to make predictions about their behavior, and we need models to help with us. And there are lots of models out there, and the choice of model depends upon what you want to ask. And one of the major things that we wanted to capture was that we wanted to capture the cell shape at a very high resolution. There are other models which like approximate cells as spheres or ellipsoids, but then they lose the information of how the shape has to change based on how it interacts mechanically with other cells. And we want to have multiple cells because we're interested in the collective motion. We want to make the cells interact with each other. We want the cells to move. And we want to ignore all the unnecessary details inside of the cell about the nucleus, about the DNA and everything. And for this purpose, we use a multi-phase field model. So in this, we're looking at the two-dimensional monolayers of cells. So these cells, they move within a two-dimensional layer. So you can ignore what happens in the third dimension. So we use a scalar field to represent each cell. So this is called as a phase field. And this phase field takes a value of +1 inside the cell and -1 outside. And then at the membrane of the cell, it varies from -1 to +1. And each cell has a phase field associated with it. And the phase field evolves as per the equation that you see here. It's a partial differential equation with an advection term that makes the cells move, and a term which utilizes energy gradients to evolve the phase field. Let's look at this in more detail. So the free energy that you saw on the right-hand side of the equation has two major components. The first one is called as a Carnot energy. And it has two terms. And if you look at the second term, which is a polynomial, it's plotted on the right-hand side, which looks like a W. And you can see that the minimum of this polynomial is at -1 and +1. That means energy is minimum at those values of the phase field. And you can see that the interior is +1 and exterior is -1. So that term is 0 in the interior and exterior, and it's non-zero only at the interface. And the first term, which is the gradient of 5, which you can see, is also non-zero only at the interface. So this whole problem is all about evolving the interface of the cells. And what happens when you use such an energy to evolve a phase field? You can see at the bottom, you have this initial condition with like a lot of random small droplets. Over time, they all coalesce into bigger droplets, and eventually you have one big circular droplet whose area is equal to the area of other droplets. The second term on the free energy makes sure that most of the domain is either the interior or the exterior, and the membrane is penalized. And the first term ensures that there are no large gradients in the field. So the field goes smoothly from the interior to the exterior. And the most important parameter here is CA. So with CA, you can control how fast a cell relaxes to a circular shape. So in a way, you can control the deformability of cells. So if CA is low, then the cells are less deformable. And if the CA is high, then you can easily like deform cells to much more complicated shapes. And the next energy is where, so the first energy that we saw, the Carnot energy, doesn't really have, it only depends on the cell I. It doesn't interact, it doesn't make cells interact with each other. But in the second term, which is this blue energy, which is the energy of repulsion and addition, we make the cells interact with each other. So why we need both of them. So this is kind of like Lennard-Jones potential, if you have heard about that. What this repulsion term does is that it makes the interior of the cell repel interior of other cells. So the red part repels the other red part. And the addition energy makes the membrane of the cell attract to the membrane of the other cell. So the interiors repel, so you don't have cell overlap, but the membranes stick to each other, like how real cells do. And essentially, like the term on the right of the integral, they are just polynomials that ensures that there's repulsion and addition. And now if you were to turn off activity or advection, and just look at how the phase field will evolve with these terms. I start with an initial condition where the cells are slightly elongated. What happens is that the tissue relaxes to some hexagonal configuration. So this is similar to how foams would relax, but it's not exactly like foams, but it looks indistinguishable from foams, how foams would relax. You can see that the cells repel each other. They are not overlapping. I also started with an initial condition that they were not overlapping in the first place. If I were to remove the interaction energy, then all of these cells would assume that the other cells don't exist and they'll just become circular. And now we put in activity. This is where things start to act much differently in the biological systems. So in biological systems, the cells move actively. They create the motion themselves. And you can see in the advection term, there's this velocity field. This field is uniform across the cell and has a constant magnitude. But its orientation, that changes with time. And we have the equation of how this orientation changes. First, you have a rotational diffusion term that causes the cell to like slightly change its orientation about its initial orientation. And then we have the other term where a cell tries to align with the direction of its elongation. This is observed in a lot of cells where cells move in the direction of their elongation. It also helps with alignment a lot. And if you put it all together, you get something like this. On the left-hand side, you can see all the cells that are moving. And if you were to remove some of the cells just to hide them, then you get the video on the right-hand side. So the right-hand side, it's just the same video, but with some of the cells that are hidden. And you can clearly appreciate like all the complex shapes that the cells are undergoing as they move across the tissue. You can see that cells are making and breaking contacts with each other. And we can track continuous shape deformations using this method. So this is something that this is like the major topic of my work. I study something called the T1 transition. This involves, so this is just a phenomenon where two cells that are neighbors. So in this situation, cells B and D, they move away from each other, they break contact, and two other cells make contact. So this is like a topological change in the neighborhood configuration. So this is called as a T1 transition. And on the bottom, you can see that you have the snapshot of a tissue, and this is the elongation of axis of the embryo that I talked about initially, where you saw that the embryo was elongating. And what you see here is some of the cells are marked, and as the tissue is elongated, the cells undergo T1 transitions. And you see this in a lot of other situations too. Like, for example, inside the lung, inside the surface of the airways of asthma patients, cells are more motile and they undergo a lot of T1 transitions there. You also see T1 transitions in passive systems where the units don't move, like foam. But you see them because there's an external force that is applied on them, and that makes the cells move relative to each other. So this is the part which some of you might be familiar with. So what I do is that we take the free energy, which is the capital F. We take the energy density due to that free energy, and we plot it. And the white dot that you see is the center of a T1 transition. So it's kind of like a symmetrically distributed point for all the four cells that change their neighbors. And we coarse grain this energy field, which you can see on the middle figure. And if you view that figure in logarithmic scale, you can immediately appreciate that this energy is concentrated at those centers of T1 transition and also at triple junctions where three junctions meet. So there's high energy at those locations. And we track how this energy evolves at those white points. And we see that what happens is that the energy slowly accumulates at those locations and then it's released extremely fast. So this is something that's been talked about quite a lot in foam systems and also in cells. But here you can easily actually track how this energy is changing. We can also look at how the cells are changing their shape. So using a parameter or like a variable called a shape index, which is the ratio of perimeter to the square root of area. This is a measure of roughly how elongated a cell is. And you can see that the cells that gain neighbors elongate differently compared to the cells that lose neighbors. So the cells are pretty much elongated and pretty much stressed when the T1 transition is occurring. The T1 transitions occur at time 0 in these figures. And as we saw using the Carney-led energy, that if cells are elongated and more curved, the energy is higher because there's a large gradient in the phase field at those locations. How much time do we have? 20 minutes. So one thing that we found, we wanted to understand the flow of the cells within the tissue, and it was very challenging because the field looks very noisy. It's hard to keep track of features in the flow field and get sensible results. So instead, we switched our approach and we started to look at flow markers. And one obvious markers are the center of masses of the cell. So you can assign a marker to the center of mass of each cell. And here we have taken a triad or a triplet of cell and attached a triangle to it. And now, as the simulation evolves, you can see that this triangle is rotating, dilating, stretching, like how a fluid element would stretch as cells move across the tissue. And we can look at some dispersion properties from these flow markers that is the center of mass. So the first obvious thing that we look at is the mean square displacement. This tells us that how much the cells move within a given time lag. We can also look at two-particle dispersion, where we look at two neighboring cells, two cells that were initially neighbor at some initial time t0. How do they deviate from each other? So we can keep track of that too. So this is the average distance of how neighboring cells move away from each other. We can also look at how the triangle area that you saw before evolves with time. So we can look at the statistical distribution of such triangles. So to illustrate again, like mean square displacement is how much those center of masses move should compared to some initial position. The mean pair separation distance is how much the sides of the triangles change over time statistically, and the mean triplets area is how much the triangles are changing over time. And here we have plotted some quantities as a function of V0. V0 is the magnitude of the advection field. So if V0 is high, the cells would definitely move faster, and V0 is low, cells would be slower. And on the top row, I have plotted the three quantities that you just saw: the mean square displacement, which is a single particle dispersion property; mean pair separation, which is pi, which is how a pair separates; and then you have the triplet separation area on the right side. And you can see that, okay, with increasing activity, the cells are moving faster and they're moving more away from each other. And there's much more higher rate of mixing as you increase the speed of the cells. But what's interesting is that when instead of plotting it against time, you plot it against the cumulative number of T1 transitions that occur. So you count when you have those T1 transitions in the tissue, when there are neighbor exchanges. And if you look at how the mean square displacement changes with T1 transitions, you don't see any specific pattern. Like, okay, the cells are all like, the different plots are all separated. But if you start plotting the mean pair separation distance, like how much the cells move away from each other, that depends on how many T1 transitions have taken place inside the tissue. So what does this mean? So like, if you have a dense system of particles and you want to see how much the particles mix or how much they move away from each other, you can track a topological feature, which is how many times they have exchanged neighbors. And so you can connect to the topology to the dynamics, and you can see a collapse. So irrespective of activity, the cells, the amount of distance cells move away from each other depends only on T1 transitions. And we see that if you look at the slope of the middle curve on the bottom side, it's for every 250 transitions, the mean pair separation distance doubles. That means it takes about 10 T1 transitions for a cell to come in between two cells. So if two cells are neighbors, and each cell must undergo at least 10 T1 transitions so that there's a new cell that comes in between. So yeah, you can, and then when we plot the square root of the triplet area, the triangle area that we saw, against the mean pair separation distance, we see that there's a linear dependence, but initially the curve is flat. You can see this also on the third figure on the top panel, that the triplet area doesn't start diverging after some time. And it's because like you need a couple of T1 transitions for the cells to separate from each other. If there's only one or two T1 transition, the cells would just become collinear. And if the cells are collinear, then the area is low, which you can see from the third figure here. When the cells are kind of collinear, even though the base of the triangle is long, the height is small, so the area is roughly the same. And now, here I've shown you, like in the second figure on the bottom panel, I showed you how the pair separation distance, like the distance between particles, changes the number of T1 transitions for change of a direction velocity. But I can change other parameters too. I can change the deformability of cells, how soft the cells are. This changes the number of T1 transitions that happens. But this dependence on how fast or how fast the cells move away from each other still remains the same. It remains the same even if I turn off diffusivity or like the ability of cells to align its elongation with its shape. And on the bottom right side, you have this figure where there's this parameter called R active. So this is the fraction of the cells that are moving. So if the R active is 0.5, that means half the cells, they're not moving. They're just passively reacting to what their neighbors are doing. And you can see something like this on the right side. There's a figure which shows the trajectory of the cells. And some cells barely move. It's most of the other cells that are doing the motion. And even in this situation, how much the cells deviate from each other is still the function of the number of T1 transitions that take place. So it doesn't matter how fast the cells are moving, whether they're soft or stiff, whether how they're moving, whether diffusing, or doesn't matter how many cells are moving. This relation is kind of universal. It only changes when we completely change the way cells move. On the left side of the bottom panel, here you can see that there are two distinct like modes of behavior, and the one with the higher slope belongs to a shear situation. In the shear situation, the cells don't move actively. You apply an external shear, a simple shear, and the cells just react to it. So there is no active motion. And in this situation, the slope is very different. As you can imagine, that in the shear situation, two cells which are parallel to the velocity field don't go away from each other, but two cells which lie at different magnitude of fields move drastically away from each other, and that creates this imbalance. The biggest difference is that in the active situation, there's no preferred direction in which cells would move. The T1 transitions are oriented randomly. So maybe the directionality is the reason why you get a different slope here. And because, I don't know, like right now we just looked at deformable cells, but maybe this is also valid for other dense systems where you just have particles which are all tightly packed to each other, and the amount of mixing or the amount of distance they move away from each other depends on how many times they change their neighbors. Maybe this extends beyond just these tissues. And here, we can kind of think as two ways in which cells move. So what we have done in this figure is we have plotted 3 different quantities as a function of V0, which is the magnitude of the advection field, but also as a function of deformability, like how soft or stiff the cells are. And the first curve is the mean square displacement, or the final value of the mean square displacement. And you see that it increases with this advection velocity, but it doesn't change because of deformability. So given a fixed value of advection velocity, all the cells are moving the same distance. But what's different is how they move. And you can see that with the middle plot, like for the low deformable situation, that is the red curve, the cells are not moving away from each other. They're moving the same amount of distance as the other cells, but they are not moving away from each other. What they're actually doing is more visible from the third figure, which shows average polar order. So this number varies from 0 to 1, and it's 0 when cells all move randomly. And if it's 1 when they all align and move in the same direction. And as you can see that in the situation where the cells are less deformable, they're not moving relative to each other, but they're moving together in small clusters. So you can decompose like the motion of particles into either with their neighbors where they don't break contacts, or into the other mode where they can actually break contacts and move away from each other. And you can use this criterion based on like when the T1 transitions occur in time and space to create directed networks. And you can, and so in this network on the left side, each node represents a T1 transition, and it is connected to another T1 transition if that T1 transition affects the other T1 transition. And now why would this be the case? Imagine that you're like in a room which is tightly packed with people, and there's just people everywhere. And there's no space to move, but you move a little bit and you change neighbors and you move away from your neighbors, but you leave your original space vacant for other cells to come in, and they would rush in to fill that space. So what would happen is that if there's a T1 transition at one location, it creates a cascade of T1 transitions close by, and the deformations can spread across the tissue, and that's visible using the figure in the middle, where I have taken multiple snapshots of the tissue, like almost 50, and overlaid on top of each other. And the color highlights the cells that are undergoing a T1 transition based on where they lie on this, based on the color where they lie on this graph. And you can see that the deformation spread from somewhere in the middle towards the sides. And to extract much more meaningful information from this, we need much longer experiments, because such data is more meaningful when you have like statistically significant data. With that, you can kind of answer questions like how long does a disturbance or a T1 transition? A T1 transition can be thought of as a disturbance in the tissue. How long does it last? And how far does it spread? So all of these questions can then be answered if you can look at the statistical properties of this graph. So this is what I'll be doing in the future. Thank you. Any questions?
Thanks, very nice.